Hi, my name is Jordan Rice, and I've been working in Dr. Robin Cooper's lab for the past 10 weeks. My summer research was possible through the Cameron program. The title of my presentation today is called Using Pharmacological and Genetic Approaches to Study Neural Circuits Involved in Mechanosensory Input. During this presentation, I will give the aim of my research, discuss why I chose Drosophila, explain briefly how synaptic transmission works. I will explain how I approached my research question from two different standpoints, using a behavioral test and neurophysiological tests. Each of these approaches could also be approached in different ways, from a pharmacological standpoint and a genetic manipulation standpoint. In the pharmacological approach, I used two different drugs, dopamine and serotonin, and to investigate the research genetically, I used two different techniques using the GAL4 UAS system and the PBAC system. The aim of my research was to develop a behavioral assay to study neural circuitry and influence of modulation on those circuits. So why did I choose the Drosophila melanogaster? The Drosophila is a popular organism used in many different types of experiments for a lot of different reasons. It has a rapid life cycle, it's easy to raise in large numbers in the lab, easy to use for genetic modifications, and its neurophysiology is similar to that of a human. And to the left is a picture of the Drosophila life cycle, and the third instar larva with the yellow star at the bottom of the picture is the stage in the larval life cycle that I chose to use in my experiments. When starting my experiments, it was important to determine the difference between a second instar and third instar larva because they do look similar in the larval stage. And to do that, I put a second instar larva underneath the microscope with the help of an advisor. And it was obvious to me that the mouth hooks were much smaller in comparison to the third instar larva and had less teeth. The third instar larva has more than 8 teeth usually, and they're very dark and much larger. The body as a whole is also larger than the second instar larva and wider. So there are many reasons to investigate this area of neuroscience, and using behavioral tests is important for many reasons. Earlier this year, President Obama called for this BRAIN initiative, basically calling for all kinds of research into the inner workings of the brain, because as one of the most important organs in our body, it's still unclear exactly how everything works. So using Drosophila to investigate the central nervous system, it's important because Drosophila display different types of mechanosensation, like hearing, proprioception, and touch sensation. And this touch sensation is essential for a range of behaviors, and that can include locomotion, foraging behavior, looking for food, and being aware of their environment and surroundings. Extracellular recordings have shown that a small touch can result in a burst of action potentials, so that is important in knowing that a small stimulation can result in this behavioral response on a cellular level. So using my behavioral assay, I hope to connect what's happening in the outward appearance and the behavioral responses and connect that to what is happening on the cellular level using the neurophysiology from the neurophysiology standpoint. Here's a picture of a third instar larva. On the top, the larva is expressing green fluorescent protein in its central nervous system. You can see the larva's brain, ventral nerve cord, and segmental nerves. And that is side by side with an anatomical comparison of the third instar larva. And you can see all the different organs and that sort of thing. Here's a brief overview of synaptic transmission. On the left of the picture, you have the cell body surrounded by dendrites with the cell axon stemming from that. and these neurons speak to one another, so to say, through this synaptic space. So what happens is a stimulus leads to firing of an action potential which travels from the cell body down the axon. This action potential activates calcium channels which allow the proteins to release vesicles which contain the neurotransmitters. These vesicles fuse to the presynaptic membrane and push the neurotransmitters out into the synaptic space. The transmitters are then bound to receptors on the postsynaptic cell. After they are released from the receptors on the postsynaptic cell, they are taken back up to the transporters on the presynaptic cell or broken down metabolically within the synaptic space. This leads me to the behavioral assay that I used in my test. After many different experiments, testing sensation and sensitivity on different parts of the third instar larva, it was obvious that the head was most sensitive than any other part of the body. So first I would use this pencil connected with clay on the top of the pencil with an insect pin pushed into the clay. And this is what I used to touch each part of the larva. So during my assay I would first touch the tail, and then touch the abdomen, and then touch the head. Between each touch I would allow at least 3 seconds and would wait for the larva to resume normal crawling. That, to me, would signify that the larva had pretty much recovered from any kind of stimulation that it had experienced. This is a video of what it looks like underneath my microscope when I would perform the hat assay. Here you can see the insect pin touching the tail, and then I'm waiting for the larva to continue crawling. Then touching the abdomen, and you can see it's pretty obvious then that the larva paused any kind of locomotion. So waiting again, and then stimulating the head. So there are different kinds of responses that I could see from each different type of touch on any of the 3 areas that I touched. This type of response, I designated a turn of less than 90 degrees. So here you can see a touch on the head, and the larva continues crawling but changes directions in an angle less than 90 degrees. This behavior response, called the tail whip, you can see a stimulation on the abdomen, and the tail flips right up towards the head very quickly like a reflex. This response was called the C-bend. Here you can see a head stimulation, and that head is pulled down towards the posterior end of the body. And for most of the 3 seconds that I allowed for each touch response, it stayed in that position. This was the most exciting response, and here you can see a brief touch on the tail, and it stimulates the larva to have this continuous rolling motion. So naturally I called this response roll. Now there are different kinds of response behaviors that you've seen so far, so it's kind of difficult to quantify what these responses might be in order to analyze them and compare them. So what I did is assign each response behavior a number, and the index that I used was based on intensity. So to me, the least intense response was no response at all. So that's when the larva was crawling, and even after touch stimulation it continued crawling, and with no hesitation it didn't seem to notice the touch at all. The next response in intensity was the pause. So you saw that one in the video a couple slides ago of the complete pausing of locomotion. The body wall contraction ceased. It was obvious that the larva felt the touch and just stopped right in its tracks. The next level had 3 different responses that shared the same numerical value. That was the turn of less than 90 degrees when the larva was crawling, hesitated for just a bit, and then continued crawling at an angle less than 90 degrees. the C-bend when it brought its head down towards the posterior end of its body, and the tail whip when it whipped its tail up briefly just like a reflex. The next intense response was a turn of greater than 90 degrees. So that's when a larva, after stimulation, would continue crawling but at an angle greater than 90 degrees. Reverse would be when the body walk contractions reverse motion for all 30 seconds of the response that I allowed. 5 was the roll, the most intense response that you saw a slide ago, or attempted roll, because in certain assays when I used drug manipulations, it was hard for the larva to control its body, so sometimes it was obvious that it was attempting to roll, but it may not have made it 360 degrees. These are frequency diagrams of the different response behaviors experienced in the different parts of the body. So in the tail, it's obvious that no response was the most frequent response. About 95% of the time, the larva did not notice any touch at all in the tail or didn't have any behavioral response to the touch. Moving to the abdomen, it's obvious that it's more sensitive in the abdomen. No response moves to only about 50% of the time. Pause is the next most frequent response, and we also have some tail whips and turns in directions and a small amount of rolls. In the head, it's obvious that the change in direction was the most frequent response. Rolls were also frequent, as well as reverses and pauses and 90-degree bends. It kind of makes sense because for you, for example, if you're walking forward and something touches the front of your body, you wouldn't necessarily keep walking into that stimulus. So that's the same in the head of the larva. So when their head was stimulated, they would turn directions, and that was the most frequent response. Whereas in the tail, when something was stimulated on the tail, of course, they just kept going forward. So that could be related to when something touches your back, you will keep walking away from that stimulus. So starting my experiments in the pharmacological approach, depending on which experiment I was doing, I could do a 1-hour acute treatment or treat the larva for 24 hours chronically. I used 2 different drug families, dopamine and serotonin. For the dopamine experiments, I used dopamine itself, L-dopa, which is the precursor to dopamine, and alpha-methylparatyrosine, which is an enzymatic inhibitor of the dopamine pathway. In the serotonin family, I used 5-hydroxytryptamine, which is serotonin itself, 5-hydroxytryptophan, which is a precursor to serotonin, and parachlorophenylalanine, which is another enzymatic inhibitor of the serotonin pathway. Methods for this assay were to prepare treatment vials. To prepare these vials, I included 0.5 gram of fly media, so normal cornmeal food, and 0.5 milliliter of solution. This solution included water and the various concentrations of the drugs that I was using. I took the larvae from the stock vials and placed them into the treatment vials very carefully to avoid any damage to the cuticle of the larva. I let them stand in the incubator in the light-dark cycle, depending on if it was a 1-hour acute treatment or a 24-hour chronic treatment. Then, after each treatment was done, I transferred all contents of the vials to a moistened 1% apple agar plate. After all the contents were on the plate, I performed the hat method and recorded the behavior responses. To analyze my data, I used a one-way ANOVA and Bonferroni statistical tests. After a few experiments, it occurred to me that it might be good to see if the larvae were actually ingesting the food that they were being placed in. Since the drugs were mixed into the food, it's important that the larva was actually ingesting the food, and that way the drug would be ingested into their body. So I placed 0.5 gram of food with 0.5 milliliter of water and put larvae into this food coloring mixture for 30 minutes. The 30-minute treatment was shorter than any of the treatments that I used in my experiments. So it was pretty clear after I removed the larvae and then looked at them under the microscope that this pretty blue color in their intestinal tract was confirmation that they did ingest the food that they were being placed in. So the dopaminergic pathway starts with tyrosine, the amino acid, which is synthesized to L-dopa, the precursor to dopamine itself. AMT is the inhibitor that I used, and that inhibits tyrosine hydroxylase, so it inhibits the formation of dopamine at this step here. Dopamine in humans is related to reward-motivated behavior, Parkinson's disease, motor control, and it is a precursor to norepinephrine. Some results from my dopamine treatments were that in the top graph you see the acute treatment. The y-axis labels are the scoring index that I mentioned earlier, with the scores of 0 to 5 assigned to each response behavior. The bar clusters are related to the tail, abdomen, and head, each section of the body that was stimulated. In the acute treatments, I used a control, a low dose, a mid-range dose, and a high dose. In the chronic treatments, I only used a control, a low dose, and the mid-range dose. I avoided using the high dose in the chronic treatments because the 260 or 100 millimolar for 24 hours was too much for the larvae and often resulted in death. In this dopamine treatment, it was seen that the tail becomes more sensitive between the control groups and the mid-range dose and high concentration. In the chronic treatments, it was seen that there was an increase in sensitivity also in the tail and the control groups in the mid-range dose. In the L-dopa treatments, the precursor to dopamine, it was seen that it decreased sensitivity in the abdomen and the head, both in the control group, between the control group and the highest concentration. In the chronic treatment, it was also seen that there was a decrease in sensitivity in the abdomen between the control group and both treatment groups. AMT, the dopaminergic enzyme inhibitor, seemed to increase sensitivity in the abdomen in the acute treatment. It's hard to give a lot of weight to this treatment group because as an enzymatic inhibitor, it's hard for AMT to really have an effect in only 1 hour. So it's safer to say what happens in the chronic treatment is more believable. And here it seemed to decrease sensitivity in the abdomen and the head. For my serotonin experiments, serotonin is synthesized from the amino acid tryptophan into 5-HTP, which I used in my experiments, and into serotonin itself. The enzymatic inhibitor I used, PCPA, inhibits tryptophan hydroxylase at this step here. Serotonin is related in humans to mood, reward, perception, anger, aggression, sexuality, memory, and attention. It's obvious that serotonin is a very pervasive neurotransmitter and has an effect on many different things in the body. From the serotonin treatments, there was no significant difference between any of the treatment groups in the acute treatment. In the chronic treatment, there is an increase in sensitivity between the control group and the mid-range concentration of 50 millimolar in the tail. In the 5-HTP treatment, the precursor to serotonin, there was no significant difference between any of the treatment groups. In the chronic treatment, there was an increase in sensitivity in the abdomen between the control group and the mid-range dose 50 millimolar. PCPA, the enzymatic inhibitor of serotonin, was not seen to have any effect in the acute treatment. In the chronic treatment, it was seen that there was a decrease in sensitivity in the head between the control group and the 50 millimolar mid-range concentration group. Approaching this from a genetic standpoint versus the pharmacological standpoint allows for different parts of the picture to be combined into one. I used 2 different systems to investigate the genetics, the GAL4-UAS system and the piggyback system. The GAL4 system which I'll explain more in depth in a second, is expressing the Shy-Beer temperature-sensitive gene. So this is a temperature-restrictive gene above 30 degrees Celsius. It's reversible, it works by depleting synaptic vesicles, and it affects the neuron as a whole. The piggyback system affects only one gene related to the neuron, so it will only affect one small part of the neuron whole. It works by an insertion mutation. The Gal4 UAS system that I use to express Shy-Beer at temperature sensitive works by transcribing the gene that encodes for dynamin. So when you cross a fly with the Gal4 line with a fly that has the UAS region with the gene that you want to express, the Gal4 binds to UAS and results in transcription of the gene that you want to express in your experiment. For my experiment, I wanted to express the Shy-Beer at temperature sensitive. So using this gene, it allowed me to impair dynamin. Dynamin is the protein that pinches vesicles and allows for endocytosis and vesicle recycling. At permissive temperatures, so at room temperature, it works and functions correctly. Vesicles are recycled and there is allowed for neural transmission. At restrictive temperatures, it impairs dynamin and you can no longer pinch the vesicles into the presynaptic membrane and you no longer have vesicle recycling. This results in transmitters that are not pushed out into the synaptic space, and this leads to no transmission. So I use this to impair transmission in this line of flies. The piggyback system works by using a transposase to cut a transposon from a plasmid and paste it into the genome. This insertion creates a disruption in a certain gene and inhibits the transcription of this gene. So you can use different markers of success, and that includes things like using the wild type that I used that had white eyes, and confirmation of the insertion would result in red eyes in the flies. This is a comparison between the mammalian serotonergic receptors and serotonergic receptors in Drosophila. You can see that they share many similar receptors, and the ones that I chose to use in my experiments for comparison are 5-HT2 and 5-HT7. To look at these receptors, I use the pback system. These pback line insertions that I mentioned that creates the mutation and stops the transcription of that gene were the pback TRH, so the insertion here highlighted in yellow, and the gene that codes for tyrosine hydroxylase results in a disruption of this gene, and so you no longer express the gene that encodes for tyrosine hydroxylase. As I mentioned earlier, tyrosine hydroxylase is the enzyme that synthesizes tyrosine into 5-HTP. The pback 5-HT2 line creates an insertion here in the gene that encodes for the 5-HT2 receptor that I showed on the previous slide. And pback 5-HT7 creates an insertion mutation on the 5-HT7 gene, so you no longer have 5-HT7 receptors. During the genetics experiments, I would cross the flies if necessary, then collect the third instars from the stock vials, transfer these larvae to the 1% apple agar plates that I moistened with DI water, and perform the hat method and record behavior responses. The results from using the pback line show that the tail had a decrease in sensitivity in the tyrosine hydroxylase knockdown and the 5-HT7 receptor knockdown. In the head, there was also a decrease in sensitivity in the 5-HT7 receptor knockdown. There were no significant differences in the abdomen. The wild-type control group used in these experiments was W1118. In summary, in the serotonin experiments, PCPA, the inhibitor, decreased sensitivity in the head. 5-HTP increased sensitivity in the abdomen, and 5-HT, which is serotonin itself, increased sensitivity in the tail. In the dopamine experiments, L-DOPA, the precursor, and AMT, the inhibitor, both decreased sensitivity in the head. They both decreased sensitivity in the abdomen, while dopamine itself increased sensitivity in the abdomen. Dopamine also increased sensitivity in the tail. So using this behavior test to investigate the responses that's happening in the outward manner, it's important to connect these outward responses to what's happening inside the larval nervous system. So moving from the behavioral test, now we can move into the neurophysiological test. This bottom picture was taken by Josh Titlow, and this larva is expressing green fluorescent protein in its nervous system. Most of the neurons seen here are sensory neurons. Here's the setup that we used to stimulate electrically the larva. On the left-hand side, you see the scope and the prep with an inserted picture that shows close up what the microscope setup was. You can see the larva dissected ventrally with the clear transparent electrode into the larva. Using the suction electrode, you can suck up this neuron that's going to this section of the muscle segments. So this neuron would innervate this whole section here. By sucking that neuron into the suction electrode, you can pick up all the activity that would be to that section. On the right here is a real picture taken from our scope, and this clear transparent tube is what is the suction electrode using to capture the activity in that neuron. Here's another picture with the larva expressing GFP, and you can see the segmental nerve with the suction electrode using to capture the activity in that neuron. Here are the recordings that we took from using the suction electrode. Every arrow indicates a touch, and you can see the burst of action potentials from what that suction electrode is capturing. So the top recording, you can see the touch, which will really lead to a burst in action potentials. Here are some more recordings. This is one taken from a stimulus on the tail. This is the stimulus from the abdomen and stimulus from the head. So you can see that just looking at one small part of the body, what the response is happening in that little spot from stimulation on different parts of the body. Instead of taking the neuron out of the larva itself into the suction electrode, you can put another electrode into an individual muscle fiber. This will allow you to see only the activity to that individual muscle fiber. Here are some recordings that you can see from the intracellular electrode. Stimulation from the tail, abdomen, and head. You can see the change in burst of action potentials. In conclusion, development of this HAT assay can be used to investigate different kinds of things in many different experiments using neural circuits. In the future, it would be nice to investigate what happens in the neural circuit during habituation. So touching a larva in one spot on its body over and over and measuring the differences in action potentials and behavior responses after multiple repetitive touches. And also, in my experiments, we only investigated what happened in touch response. There are many different neural circuits that you can investigate, like olfaction, sight, hearing, and it would be neat to see what's happening in the nervous system in those neural circuits. I would like to thank Dr. Robin Cooper for working with me for the past 10 weeks, being very patient. He was a great mentor to work with, eager to teach, and very fun to work for. Josh Titlow and Zana were the grad students working under Dr. Cooper, and I worked very closely with them during my experiments. Everyone else in Cooper Lab made my experience fun and enjoyable, and I'm glad that I got to meet everyone that I did in this lab. And I would, last but not least, like to thank the Cabrin program for giving me this opportunity to complete this research and be exposed to all kinds of things that I had never been exposed to before. In closing, thank you for watching this presentation. I will welcome any suggestions or questions that you might have, so feel free to comment below. Thank you.